The hypoxia inducible factor is a transcription factor consisting of an alpha and a beta subunit. One of 3 different alpha subunits dimerizes with one of 2 different beta subunits. The alpha subunits are sensitive to oxygen, whereas the beta subunits are not. Among the alpha subunits, the function of HIF1 alpha and HIF2 alpha are best understood. Here, different domains of the HIF1 alpha subunit are shown. The subunit contains an N-terminal basic helix-loop-helix motif, which associates with the DNA. Near the C-terminus, 2 transactivation domains can be distinguished. These domains guide the transcription of the HIF1A gene into mRNA. In addition to the helix-loop-helix domain, an adjacent domain is essential for dimerization with an HIF beta subunit. The proline residues are crucial for protein stability, and the arginine residue regulates transcriptional activity. The HIF2 alpha subunit contains domains similar to those in HIF1 alpha. The HIF1 beta subunit contains the basic helix-loop-helix domain, which is part of the dimerization motif. The protein contains only one transactivation domain. As mentioned before, HIF alpha subunits contain 2 proline residues, which are critical to the stability of the proteins. For simplicity, the animation here shows 1 instead of 2 residues. Normally, the concentration of molecular oxygen in most human tissues is in the range of 10 to 30 micromolar. Under these normoxic conditions, one or both of the critical proline residues in HIF alpha proteins will be hydroxylated by a member of the prolyl hydroxylase domain family, abbreviated PhD. PhD2 is the major hydroxylase, which hydroxylates HIF1 alpha. The PhDs are dioxygenases. These are enzymes that incorporate both atoms of molecular oxygen into their products. Prolyl residues are the substrates of PhD dioxygenases. One oxygen atom is used in the oxidative decarboxylation of alpha-ketoglutarate, an intermediate of the tricarboxylic acid cycle. PhD dioxygenases require bivalent iron. The products of the reaction are a hydroxyprolyl residue, carbon dioxide, and succinate, also an intermediate of the tricarboxylic acid cycle. When one or both of the critical proline residues have been hydroxylated, the von Hippel Lindau protein, abbreviated pVHL, binds to HIF alpha. The VHL gene is a tumor suppressor gene. Inactivating germline mutations of the gene predispose individuals to tumors such as clear cell renal carcinomas. Biallelic VHL inactivation is found in approximately half of all sporadic clear cell renal carcinomas. The von Hippel Lindau protein associates with the ubiquitin ligase complex containing a longin, culin, and ringbox protein 1. An E2 ubiquitin conjugating enzyme attaches ubiquitin to HIF alpha. Polyubiquitinated HIF alpha is degraded by the 26S proteasome. In addition to the proline residues, HIF alpha proteins contain a critical arginine residue in the C-terminal transactivation domain. The residue is hydroxylated by a dioxygenase called factor-inhibiting HIF1 alpha, or FIH1. Similar to the PhDs, FIH1 incorporates both atoms of molecular oxygen into its product. One oxygen atom is used in the oxidative decarboxylation of alpha-ketoglutarate. The products of the reaction are hydroxyarginyl residue, carbon dioxide, and succinate. The hydroxylation of the arginine residue inhibits the binding of the P300 and CBP transcriptional coactivators. Under hypoxic conditions, HIF-alpha proteins are no longer hydroxylated by PhD dioxygenases. A HIF-alpha subunit associates with a HIF-beta subunit and binds to consensus sequences in the DNA. The P300 and CBP coactivators bind to HIF-alpha. The complex activates the transcription of target genes.